do you have? Okay. So check in. Don't forget to check in on my clicker. Yeah, that's what I thought. So. Look, I'm not so bad. Exams are back and case studies back. And y'all, you don't have to write the questions for the case studies. Like a bunch, like it was like a half and half. Half the people did and half the people didn't. Like you don't, if you want to, cool. Like I'm not gonna, but you could save time and just not write the questions for the case studies. It's cool. I know what the questions are. So this is just the first case study? Just the first case study? I know, right? I had the whole break. It's just the first case study. You can expect the second one next week. I'm working on it. I'm getting there. I graded for your class, for this class for a long time. Oh, you should see, I didn't realize that I hadn't formatted our grade book for the year yet, for the semester yet. So the grade book on Moodle is now like percentage-wise the right things, everything's in there. So it looks like a normal grade book should, instead of like, I don't know, I opened it up and it was like 1900 points and some weird crap. And I was like, no. I just hadn't realized that I didn't fix that yet. Okay, so questions on exams. Exams are up here if you want to grab your exam and case study stuff. So all the grades that you have back now are on Moodle, all your homeworks, all your reading quizzes, everything I have for you so far, except for the second case study, is posted. I'm sorry it took me so long. It happens. Any questions before we start?
Extra credit? Any extra credit?
No. But there was a curve.
Oh, that doesn't make me feel better.
There was a curve. There was a 9-point curve.
Oh, that does not make me feel better. Doesn't.
Questions? Yeah, eventually, before not the case study. The case study is yours. But the exams eventually, before you walk out the door, just put them back on the desk is cool. But I'm happy to answer any questions or go over stuff, you know, at the end of class if y'all have questions. I tried to write a lot. I didn't necessarily take off a whole lot of points, but I tried to write y'all a lot just so that you would know a little better what I was looking for in exam too. Yeah. Oh, the one thing I did want to tell y'all is that when you're taking an exam, you can ask me, like, am I answering the question? Because I think a couple of times, a few of y'all went down a path that wasn't the path of the question. So you can absolutely raise your hand and say, am I going about this the right way? I'm not going to tell you if your answer is right or wrong, but I am going to go, you're barking up the wrong tree. You know, like you need to kind of think about X or Y, you know? So always ask. The worst thing I can say is I can't answer that. Okay, good. All right, I'm trying to think what else. I wrote myself a few notes. I did want to, let's see, too many folders, too many papers. There were a few that I wanted to, what was it? Oh, when y'all were doing the, what does the pentose phosphate pathway accomplish for the cell? A bunch of y'all picked like the 3 different states of the cell, right? Which 2 of them worked. And I started to take off points, but I didn't. So if you're asking, what does the pentose phosphate pathway do for the cell? And one of the things you talk about is a low ATP state. Is the pentose phosphate pathway on? Is it doing anything for the cell? No. It's a way to regulate it, absolutely. So if I ask you about regulation, abso-freaking-lutely. But just be careful that what you are answering is not just, oh, I saw that in the, you know, in a figure, or, you know, whatever it is. Just make sure that you're thinking about the question and you're answering the question. I think that was a good example of that. All right. Yeah. Anything else? Good. Okay, cool. So chapter 15, we're talking about lipids, right? So I think we ended right here, right? We were talking about sphingolipids, right? And so when we talk about sphingolipids, sphingolipids are different than triacylglycerides, right? What's the backbone of a triacylglyceride? Glycerol, right? In sphingolipids, we no longer have a glycerol molecule as the background. We have sphingosine as the background. And then we add to that sphingosine a fatty acid, and then we add other stuff. So let's look at what happens. Here's our first one. Let's see if I can get my pen rolling today. So this blue part of ceramide here, this is actually sphingosine. G-O-S, sphingosine, right? And so what we did to sphingosine is we took sphingosine and we added one fatty acid, and we now have what's called ceramide. Now, we don't actually have a sphingosine, a sphingomyelin yet, right? So what we're going to do is then we're going to add head groups, just like we do on a triacylglyceride to get a phospholipid. We're going to add our head groups, but we're going to add the head groups to sphingosine. So what do we have here? A phosphate, and if it's phosphocholine, it's a phosphate and choline, right? And so now we've made sphingomyelin. So sphingomyelin is actually an example of a sphingo, H-I-N-G, a sphingo phospho, P-H-O-S-P, phospholipid, right? Okay, now we can take that core of sphingomyelin and we can change what we add as our head group, right? This was our phosphate, P-H-O-S-P, phosphate, choline, right? And we can say, okay, well, instead of adding a phosphate and something else, I can start adding, what are these? What are these things? They're sugars. So what do you see at the end of the name of the lipids that contain sugars? Sides. Those are sides, right? And that's telling me in the name that they contain sugars. You could contain one or a few sugars, or you could contain many sugars. So these two are not sphingospholipids. What are they? They are sphingoglycolipids. And I can tell just by the name which ones are which. Now, how important is the arrangement and the type of sugar that is attached to these lipids? Well, extremely important, extremely important. And this is something that you might have heard about in our community. You heard about Tay Sachs disease? Yeah, so this is something that that is a disorder that was found in like the Cajun population. And so when you look at these gangliosides, right, you're looking at particular sugar groups. So watch, I'm going to skip ahead and then I'm going to come back. So here's a ganglioside, right? We have ceramide as our base instead of a glycerol molecule, right? And we have our, gives you two fatty acid tails because ceramide had one and then we added another fatty acid to it. Then we're adding these sugar head groups. So initially a ganglioside is made and then basically cut down. And so you have an enzyme in you that is hexamidase A, and this one will actually cleave this N-acetylgalactosamine off of the precursor ganglioside. Then you're going to make a functional ganglioside, right? So in people who have Tay Sachs, the problem is not that you aren't making this final product, because you aren't, right? So this is lost. That's true. But the big problem is that this precursor molecule, the GM2, this one actually piles up where it's not supposed to. So it has aberrant accumulation. And can anybody guess where it accumulates? So it accumulates not in the liver, in the spleen and the brain. So if you have something piling up, this GM2 piling up in the brain, what kind of physical symptoms do you think you're going to have? Disorientation. You're going to have loss. It's going to be something cognitive. It is. And people who have Tay Sachs, they typically don't live very long. Usually 6 or so is kind of the average lifespan. And what you see with these children is that they like lose themselves, and it's horrible. It's terrible. They go from having personalities and things like this into losing all of that. And I had a thing for us to read about it, but my computer wasn't working this morning, but I posted it to Moodle. So maybe if we get to it at the end of class, we can pull it up. It is genetic. It is genetic. And so I posted one PDF, and it's a news article, and then attached to the news article is like one of the very first papers that was published on Tay Sachs disease in the Cajun population and the genetics of the disorder. And so it is a recessive disorder where you have to have 2 mutations. And so basically, you know how when the Acadians came down, it was a small population of Acadians, right? So in that small population of Acadians, there was more interbreeding than should have been, right? And so, I mean, it happens with a small population. It just does. And so the disorder became more prevalent because of that. And so now, I mean, now we know how to screen for it and things like that, but it's definitely something that impacted the population, right? So this is not the only disease where you have a malfunction in the regulation of how these gangliosides are made. You can have 2 other diseases where you have that same thing. You have a precursor molecule. The precursor molecule is supposed to be cleaved by an enzyme. You're supposed to make like the final product, the useful lipid inside of, usually it's the brain, but you don't. And so because you're missing, or you have a mutation in the enzyme, whichever one it is, then you have a different disorder, and it gives you slightly different symptoms and that kind of stuff. But the Tay Sachs is what's really, I guess, kind of relevant to our community. Okay, so we've talked so far, really, right? We started talking about fatty acids that are linked to a glycerol, and then have a phosphate and a polar head group. So what do we call these? Phospholipids, right? So these phospholipids, if you can have the same basic head groups, you can also put that onto a sphingosine molecule and attach your fatty acid, and then you have a sphingolipid, or you can change that head group into glycan groups. But what we haven't talked about, which we're going to talk about a lot today, is this lipid called cholesterol, right? And so we know that we have different concentrations and different distributions of these different lipids within the cell membrane, depending on what the function of that part of the membrane is. So when you look, you even have a different distribution of the types of lipids in the part of the membrane that faces the outside of the cell versus the part of the membrane that faces the inside of the cell. So a lot of times what you're going to see are the gangliosides on the outer portion. Why? Well, so usually they're used in some kind of cell recognition. cell signaling, something like that, because these long sugar groups are protruding out into the extracellular matrix and all that kind of stuff, and they can interact. But what about your cholesterol? Does your cholesterol stick out in any way, shape, or form? No, not really. Really, it's embedded within the membrane. What does cholesterol really do for your membrane? It controls the fluidity or the rigidity of the membrane. That's exactly right. Okay. So if you look at a breakdown of the plasma membrane, what we've kind of been talking about is just lipids, but is that all that's in a membrane? Absolutely not. There's tons of other stuff, tons of other stuff. So if you look at the distribution, see the blue versus the yellow, what's the difference between the two? What's the blue? They're all what? They're all proteins. What's the yellow? Lipids. What do you have more of? Proteins. You actually have more of proteins. Basically, you have a lot of things that your cell membrane is doing for you. And proteins are accomplishing those changes, right? So you can have transport proteins. That's your biggest one. You have to get things in and out. So that's going to be your biggest one. Then you have things that are involved in metabolism, so breakdown or biosynthesis, things like that. And then if you look at your lipids here, what is your most prevalent lipid inside of the lipid bilayer? Glycerophospholipids. So that's kind of your bulk. And then you have different phospholipids to accomplish other tasks within the plasma membrane. So I want to show you, this is like one of my favorite videos. I can make this play. The inner life of the cell. So this was made by Harvard. What's that? I might have, but like different parts of it.
Yeah.
I think I showed from this, I think from last semester, I showed the myosin walking along the actin and dragging the vesicle behind it. So I'm going to start at the very beginning, and it shows you a little bit, and then it jumps right into the membrane. You should like watch the whole thing, but oh, this is the short one. I wanted the long one. Where's the long one? Wait. That's the wrong one. The long one's like 8 minutes long. You want the long one. I'm gonna have to update that link. Here one, full version narrated. This is the one you want. Okay, so they do all this. Are we getting sound? I don't know. So they do all this on the basis of a white blood cell that's traveling through a blood vessel. And so all of the cellular stuff that they're showing is really in a white blood cell, but like it all kind of translates to other cells. Okay, now cross your fingers the sound works.
Leukocytes roll slowly on endothelial cells. These selected endothelial
Louder,
cells
softer.
interact with ESPN1, a glycoprotein
Oh,
in
good.
leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell types.
So what's the extracellular matrix? Do you see all this like spaghetti looking stuff? That's all extracellular matrix. So you have some cells that are embedded inside of extracellular matrix and can't move, but then you also have white blood cells that have some of that on the outside, even though they are moving in your bloodstream.
The outer leaflet of the lipid bilayer is enriched in sphingolipids and phosphatidylcholine. Sphingolipid-rich rafts raised above the rest of the leaflet recruit specific membrane proteins. Rafts' rigidity is caused by the tight packing of cholesterol molecules against the straight sphingolipids hydrocarbon chains. Outside the rafts, kinks in unsaturated hydrocarbon chains and lower cholesterol concentration result in increased fluidity.
Okay, so let's go over that one time, right? This is a raft. What's in the raft? So cholesterol is in there, and then what kind of lipid? What kind of other lipid is a lipid? The sphingolipids, right? So the sphingolipids, look at the difference in the tail. It's hard to see because I've paused it. But do you see how the tails are like straight like a book? Why did we say, what's the biological effect of having those really straight fatty acid chains? Tell me about London dispersion forces. More, right? This is like that perfect book where the pages like perfectly one on top of the other, right? So that's the lipid raft. So you not only have cholesterol, a high concentration of cholesterol, but you also have what do we call those that are that way? We call them the straight saturated, right? Saturated and unsaturated. The ones over here are the phospholipids, right? The phospholipids tend to be unsaturated and so end up having those kinks, and so they don't pack together quite as tightly. So remember when you were thinking about the fats in your kitchen, the butter is saturated fat. So what's its state of matter at room temperature? It's a solid, right? How about oil? Oil has more unsaturated and it is therefore a liquid. This is the same thing in your plasma membrane, exactly the same thing. We have a lipid raft that has very what? Saturated straight chains that have lots of London dispersion forces, and it has lots of cholesterol. So you have this like very dense, it's not really moving very much. And that's why they call it a lipid raft, because the raft itself can move, but the conformation and the composition of the raft will not change. That makes sense? And then you usually have certain proteins that are embedded inside of that raft and do certain things. Isn't that cool?
In sites of inflammation, secreted chemokines bound to pepper and sulfate covering of glycan endothelial cells are presented to leukocyte 7 transmembrane receptors. The binding stimulates leukocytes and triggers an intracellular cascade of signaling reactions. The inner leaflet of the bilayer is a very different composition than that of the outer leaflet.
So you hear that? The outer leaflet, the inner leaflet, that the outer leaflet has one composition and the inner has another composition. And that's exactly what we saw in the diagram that was in the slides, right? That you had a different composition of phospholipids on the top than you did on the bottom. But you can still see, did you see the bottom of the raft, right? You still see the bottom of the raft.
While some proteins traverse the membrane, others are either anchored into the inner leaflet by covalently attached fatty acid chains, or are recruited through non-covalent interactions with membrane proteins. The membrane-bound protein complexes are critical for the transmission of signals across the plasma membrane. Beneath the lipid bilayer, spectrum tetramers arranged.
So then it goes on and on and on about organization, and you should go and watch it because it's freaking awesome. If we have time at the end, I'll play it. But it's really cool. But I thought that gives you a little bit of an idea of what we're talking about when we talk about fluidity, right? You can see how much change and movement you had in the phospholipids, but you didn't have so much in the sphingolipids that were in the lipid rafts, right? So just kind of gives you that visualization. All right. And what portion did we say of the membrane is our phospholipids? A large portion of it, right? Most of it is. And then you have the other components that do specific jobs like cholesterol, maintaining fluidity, all that kinds of stuff, right? So when we talk about those lipid rafts, right? Not only did you have a different kind of lipid inside of the lipid raft, but what else do you have that's different inside of that raft? Proteins, right? A particular kind of protein, depending on what the lipid raft is, but it's very densely packed, right? And a lot of times they have these huge transmembrane proteins that span the entire membrane. So you remember you could see the bottom of the raft and you could see some bottom part of that protein that span the entire membrane. So sometimes they're receptors. sometimes you want a collection of those receptors in a certain spot on the plasma membrane, and they can be kind of shuttled around to a particular place on the membrane. Some are in lipid rafts and some are not. Because they're such a huge class, there are some that are and there are some that aren't. Yeah. All right, so what have we talked about in general for lipid functioning, right? We talked about lipids functioning as, in the very beginning of the chapter, functioning as what? Energy storage, right? That's the number one thing that most people think about, fats, energy storage, right? But generally we say lipids. Okay, what was the second one that we just finished? Cell structure, right? Cell structure, membrane structure. Now we're going to talk about a third function, and that is cell signaling. So when we talk about cell signaling, you could just put yourself a big asterisk star. This is going to be cholesterol. The bulk of cell signaling comes from cholesterol, not all, but most of it. So when we talk about things that are cholesterol, right? Just like fatty acids, just like triacylglycerides, just like sphingolipids, right? They're going to be hydrophobic. And it's that hydrophobic ability that allows them to interact very, very, very specifically with receptor proteins, right? We know that that interaction between the ligand and the receptor has to be super, super, super sensitive. So what we're going to see in our structure of all of these cholesterol-derived cell signaling molecules is that everything basically has that core of a cholesterol, and then we start modifying side groups just a little bit, and we get very different substances and very different signaling. So this is our precursor cholesterol, right? We know that cholesterol in its normal form gives us 25 to 40% of all the lipids inside of our plasma membrane. Now, that's a different percentage than what you saw in that pie chart. Why? That pie chart was both lipids and proteins, right? So if you made the orange part 100%, then of that orange, 25 to 40% would be the cholesterol, right? So don't get confused on the numbers there. All right. So you have carbons in these four rings, and then you typically have a short fatty acid tail. Typically, it's not very long. And this is what controls that fluidity. This is one of the things that controls the fluidity. So we can take this cholesterol molecule and we can alter it. And do you see how we've maintained this nucleus? That's what they kind of call it, the cholesterol nucleus, where we still have these four ring structures, but what you do to the tail and what you add to the rings will change the properties. So you can make things that are steroid hormones that will be highly involved in cell signaling. And we'll talk about quite a few different steroid hormones. But you could also make things like bile acid, right? What does bile acid do? Yeah, it's fats. So this is what your bile is. Remember, we talked about your gallbladder, and that's what's in your gallbladder. Well, if you have one, you know, for those of us who don't, it's okay. We still make it in the liver. We just don't store it. It's okay. All right. So it's a metabolic precursor to steroid hormones. That's what we said, and to bile acids. The cholesterol is derived from melavonate, and we'll look at some structures here in just a minute. But the thing that's kind of important to know is that if we have too much cholesterol, right? I know you've heard this, it's going to lead to cardiovascular disease, right? So how do we transport cholesterol? This is something we did before. How do we transport it? What do we transport it in the what? And they can be in different ones. Choliomicrons is absolutely one, but in the lipoproteins, choliomicron, CHOL, Y'all know what I'm talking about. If I get the spelling wrong. Right? And so what's the problem there? If we're transporting them all around the body, how is that going to lead to cardiovascular disease? What are we going to end up depositing them in? Places they're not supposed to be, right? So what do we call that when you have deposits of lipids on the inside of your blood vessels? Plaque, atherosclerosis, right? Leads to accumulation in blood vessels. You get plaques, atherosclerosis. So if you have high cholesterol, you go to, let's say you go to the doctor, they tell you you have high cholesterol. What do they do? They prescribe something usually. They prescribe a statin, and we're actually going to talk about statins later, but we'll just say in general drugs, right? So what does the drug do? It prevents the making of cholesterol. So then the only cholesterol that you have in your body is what you consume. So then you can regulate the amount of cholesterol in your body that way, right? Usually through diet. So usually there's a drug that slows, you don't stop 100%, but slows cholesterol synthesis, and you control cholesterol intake, right? And then you can regulate the overall amount of cholesterol inside of your body at any given time. And we'll actually talk about statins later. Okay, good. And we already did this. I don't know why they duplicated that. Yeah.
So it only becomes a problem like when it's transported is when you have too much cholesterol.
Typically, yeah.
So it's okay, like it has to.
Well, you have to have some. If you don't have any cholesterol, are you going to have lipid rafts? No. How's your membrane going to be? It's going to suck, right? It's anything in excess is not good, right? Anything. So whenever you're thinking about metabolism and that kind of thing, something's not enough and something's too much. There's where you're going to have defects and problems and all that kind of stuff. You need an amount of basically everything. It's just, are you getting too much or are you getting not enough? Right. Okay. And we talked about this, right? We did this. Okay. So we're first going to talk about hormones, right? Steroid hormones. So when we talk about steroid hormones, why are these going to be different in terms of cell signaling? You're going to have to think back to Biochem 1. Most ligand and receptor signaling occurs where? Cell membrane, right? But tell me about cholesterol. It's very what? Non-polar, and it can just slip through the membrane. So are you going to have signaling at the membrane? No. Where's it going? It's going to the nucleus, and it's going to interact with receptors. But what do we call these particular receptors that are inside the nucleus? Nuclear receptors. What's another word? You don't ever call them nuclear receptors. What do you call them? Transcription factors. No one calls them nuclear receptor proteins. That's what they are. It's absolutely what they are, but nobody calls them that. So because they're transcription factors, now it makes sense, right, to know that they're altering expression for specific genes. That makes sense. So we categorize them by their function. You have glucocorticoids, you have mineral corticoids, you have androgens, you have estrogens, right? Some of these probably sound a little bit familiar to you, right? But think about how important these groups of steroid hormones are. I mean, cell development of, you know, an entire human being depends highly on hormones, right? So when you think about these kinds of hormones, a lot of times these are acting in a very distant fashion. What do we call that? They're produced one place in the body and they act on something different. Like maybe it's produced a hormone that's produced in the brain and it travels to the ovaries or something. So remember paracrine? You remember all that? Yeah. Endocrine, paracrine, autocrine. So autocrine does self. This is how I kind of like to go about it. It narrows it down, right? So autocrine is self. Then what? Paracrine is close by, and then endocrine is far away. Okay. Right. Okay. So if you look, do you have to memorize the pathway? You do not have to memorize the pathway. Please do not memorize the pathway, but you do have to know kind of what the general functions of the different hormones are, right? So here's our cholesterol, right? The ABCD is our cholesterol or our steroid nucleus, right? And so what you end up going through are a series of different reactions that basically add or take things away from that steroid nucleus. So our simplest form is progesterone, right? One of the simplest. Then we can go a couple of different ways. You can go the corticoid pathway and go to a corticosterone. You could go to aldosterone. You could go to cortisol. So all of those are involved in the inflammatory response and that kind of thing. And we'll talk about that in a minute. Then you could go down and you can make testosterone, right? And who has testosterone? Just males? Everybody. Males have more testosterone, but everybody has testosterone. And then testosterone can be altered and we can turn it into estradiol, right? That's one of those that's in regulating the monthly cycle, right? So here's kind of the ones you have to know. So progesterone, the Corpus luteum. So this one is a hormone that's produced inside of the ovaries. So every month this hormone is made. And when you don't become pregnant, then it stops making it. So it's like the prepare to be pregnant hormone. And then when it realizes, oh, it's not happening this month, then it shuts off, right? Cortisol. A lot of people hear the cortisol is the stress hormone, right? You've heard that before. So you make it in the adrenal cortex. Where's the adrenal cortex? In the adrenal gland, what is the cortex? So you have the medulla, which is the middle, the cortex. No. The cortex is the outside. Yeah. Medulla is the middle. Yeah. Yeah. Aldosterone, which is again made in the adrenal cortex. So the adrenal gland is not just, people think adrenal gland, it's just like fight or flight, stress, that kind of thing. But that's not the only thing that your adrenal cortex will do for you. It's also really, really important in maintenance of ions in the kidneys. Now, if you're going to change the concentration of ions in the kidneys, what are you inherently regulating? Osmoregularity, right? Well, where is fluid and ions? What's the kidney doing? It's filtering what? Blood to make urine. So you're really regulating blood pressure via the kidneys, via the adrenal gland, which is kind of cool. All right, testosterone, both the testes and the adrenal cortex. So that's male reproductive organs. Then estrogen in the ovaries for female reproductive organs. So some of these you probably already know. So here's the thing that we can do as chemists. This is the interesting part. You have, for example, a natural androgen that the body is going to make in order to promote male reproductive development. the drug manufacturers have a plethora of targets that they can use in drug development, a plethora. So here's an example of one. This is a synthetic androgen that mimics this natural hormone. So what's the difference? Yeah, you took that branch and you made a double bond. So a lot of times this is a hormone that bodybuilders will use. So if this is an agonist, what is an agonist? Right? So if this hormone promotes reproductive development, this hormone will speed it up even more, right? So an agonist, it increases the natural signaling, right? Well, just like you can manipulate natural signaling, you can also block natural signaling, right? An antagonist. So we have on the very last one, and I'm not even going to try to pronounce it, but this last one is an antagonist for an androgen. And so this one is used to treat prostate cancer. So if you have an antagonist, what does an antagonist do? Blocks natural signaling. Right? So this will slow tumor growth. So you're going to see in the pharmaceutical industry that they're going to use either side of this. And you see a lot of this in steroids, right? So if you have asthma, what do you take? An albuterol inhaler, which is going to be a what? A bronchodilator. It's a what? It's an antagonist because it's going to stop the inflammatory response. Sure.
So like as prevalent as prostate cancer is, why would everybody just get one of these longer after they finish puberty? Because to understand that age doesn't matter after puberty, so why not just be proactive and stop prostate cancer on water?
I don't know if there are any studies about long-term use of it. Like, if you're going to put somebody on that for years, right? That's the same thing that they thought about. Why don't you just put all females on hormone replacement therapy as soon as they go through menopause, right? Then you won't have hot flashes, you want this, you want that. Well, then you get cancer, right? So you have to be very careful about putting anybody on long-term anything. So I don't know the research behind it. So I don't know, but long-term use is tricky. Okay. I don't know where we were going with that. Vitamin D. All right, vitamin D. Vitamin D is really cool, right? So what happens if you don't see sunlight for a long time? What do you get? Vitamin D deficiency, which leads to, well, not right away. Y'all know Tiny Tim? Remember Tiny Tim? Rickets. You get rickets, right? So if you've ever seen children in third world countries who don't get dietary vitamin D, have you seen their legs? Their legs end up looking like bowed, right? Because they end up not having enough vitamin D in their systems, and so their bones don't grow properly and all that kind of stuff. But how do you get your daily allotment of vitamin D? What do? Just go out in the sun, right? But what do we have now in the grocery stores? Vitamin D supplements, but also, have you seen fortified listed on some of the foods and things? Milk. What does that mean? What does fortified mean? Pump full of what? Pump full of vitamin D.
Well, I wanted to say
Vitamins. Vitamin D. Yeah. So that we don't have that in our developed nations. A lot of times you're going to see it in people. It was a big thing in Canada not that long ago. But you got to think Canadians, right? They go out in the sun as much as the rest of us. No, it's too cold. Oh my gosh, I couldn't imagine that. Lived in Iowa for a short time. That was plenty cold enough for me. As far north as I want to go. All right. So now we're kind of going to change gears a little bit, right? Everything we've talked about so far was endocrine. Endo, E-N-D, endo, C-R-I. Endocrine, right? So tell me about endocrine. Acts from site far away, right? A distant site, that is correct. Okay, so now there are going to be lipids that are involved in paracrine actions, right? But they're going to have a different composition. So our endocrine was our cholesterol, CHO, cholesterol-based lipids, right? They're going to last a long time. They can travel from the brain to whatever organ, or the adrenal gland to the kidneys, or whatever it is. But paracrine are basically going to be fatty acid based. And they're not going to last long. So this is a short distance. And they're not going to last long. So they're not long lasting. Right? Have half-lives of only a few minutes. That's not very long at all. And this group, we call them the icosanoids. You don't say the E. The E is silent. Icosanoids. So they're basically unsaturated fatty acids, and they're hooked to the membrane, right? And so how do you initiate signaling of things that are hooked to the membrane? They can't go and travel anywhere until you cleave them off the membrane. So they're going to be cleaved, right? Then they can travel inside of the cell, and usually they're modified by mitochondrial enzymes. And they're produced and act at that same cell, basically. So you have four major classes. You have the prostaglandins, right? So what are some things that prostaglandins might do? You've heard that before, prostaglandins? Yeah, so this is smooth muscle regulation. You also have inflammatory response. Prostacyclins are a little bit different. These will also control smooth muscle as well, vasoconstriction, vasodilation. Okay, how about this? Throm, what does that refer to? Right, a thrombus, what's a thrombus? A blood clot, right? So these are going to actually help to regulate blood vessels in terms of constriction. So this is blood vessel constriction. Yeah. Yeah. What was the other one? I don't remember. Leukotrienes. Leukotrienes, these are inflammatory resistors. T-O-R, inflammatory, right? So we'll say mediators, because they can control it either way. And they can also, and a lot of these can also control smooth muscle contraction as well. So a lot of times you're going to see smooth muscle contraction in all of your major classes of icosanoids, if I could say it. So the drug industry, of course, is going to take advantage of what we know about our lipids and our icosanoids. So, which one? Oh, coumarin, that's right. It's fine. All right, so what normally happens? Do you remember how we said you're going to have one of these icosanoids linked to the membrane and you need to cleave it? So do you remember a signaling pathway that we talked about before that would actually cleave a lipid off of the lipid bilayer? Yes, right? A GPCR. So when we have GPCR activation, the G beta gamma disassociates from the G alpha. So, right, remember, normally they're all hooked together. And then when you activate them, the beta and gamma subunits separate. And when the beta gamma subunits separate, ooh, too many S's today, then you're going to activate an enzyme. What enzyme do you activate? PLC, generally the class of PLC. But in icosanoids, it is phospholipase A2 that gets activated. So it's a different phospholipase.
Would that be the second messenger?
No. No. But you're on the right path. But you don't have to classify them that way. Okay, so we're going to use phospholipase A to cleave, and we're going to release a part of the fatty acid that was linked to the membrane. We're going to release arachidonate. Arachidonate here is our fatty acid. So why do we call it arachidonate if it's a fatty acid? Because we've lost the hydrogen, right, that makes it a fatty acid. So now it's the conjugate, right? So we call it arachidonate. Okay. So now arachidonate is actually going to be processed and turned into things that will activate our inflammatory response, right? So swelling, redness, right? All the things that you can associate with an inflammatory response, right? So basically what you're doing down here is creating an inflammatory but you have to have some enzymes that are going to process arachidonate into our final effector molecules. So two of those are COX-1 and COX-2. So this is what aspirin and ibuprofen work on in order to reduce the inflammatory response. They inhibit COX-1 and COX-2. If you inhibit the enzyme that will process them into the effector molecules, what are you doing? Stopping the inflammatory response, right? You have other classes of drugs that will inhibit COX, not COX-1, but COX-2, and then another of the prostacyclin synthases. So ones that make those ring structures. And if you do this, it's not, you have to be very, very careful because you can actually increase your risk of cardiovascular disease if you use these COX-2 and prostacyclin synthase because they can actually interfere with some of the derivatives that you're going to need for heart health. So you have to kind of be careful about that. But what's the other symptom of taking aspirin? Stomach, right? Okay, so why is that? Well, the COX-1 and COX-2 enzymes are also responsible for producing that mucus layer, the signaling pathway that's required to make that mucus layer. So if you have a lower production of that mucus layer and you're on aspirin, ibuprofen, right? Ibuprofen for a long time, what are you actually going to end up? Yeah, and usually you end up having ulcers, like ulcers. Yeah, where your stomach is basically like eating itself, which is.
Somebody has like an autoimmune issue, where they're constantly making that inflammatory response.
Yeah.
One of the ways to help that is aspirin and ibuprofen.
Yes, absolutely, absolutely. But you have to be careful about taking it long term, right, for your stomach health. But then you could take a different class, but then you have to worry about your cardiovascular
So
health.
what if the inflammatory response is in your stomach?
What if it is in your stomach?
What if it's like in your stomach and gastrointestinal system?
Then I'd be careful taking NSAIDs. I'd be careful
You'd be
taking.
screwed, basically.
Yeah, I'd be
Right.
careful. Well, yeah,
Think
there
about it.
there are some problems with that, right? There are some problems. All right, so that is everything that I have for the end of, right? Is that the end? That's the end. Yay, that's the end. for the end of lipids, right? Lipid structure and function. Next time, we're going to talk about lipid metabolism. So I have posted on Moodle. I really want y'all to read it, but I couldn't print it today. So on Moodle, if I could just see Moodle. Okay, right here, the Tay Sachs Disease handouts. I had a little activity that went with it, but I can't print it, so it's okay. And apparently the internet doesn't like me today anyway. But it's a really interesting read. If you just kind of want to understand what Tay Sachs is and kind of how it affects the families and things like that. So if you want to read that, I'll make that optional. Y'all want to see the rest of the inner life of the cell? It's so cool. Okay. Do y'all want to see it from the beginning, or you want to?
We have time for both.
Yeah, we have time for both. It's kind of nice to see it from the beginning because, like, we'll have talked about lots of these different things. So we'll let it play from the beginning, and then I'll let you be free for the day.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell types. The outer leaflet of the lipid bilayer is enriched in sphingolipids and phosphatidylcholine. Sphingolipid-rich rafts raised above the rest of the leaflet recruit specific membrane proteins. Rafts rigidity is caused by the tight packing of cholesterol molecules against the straight sphingolipids hydrocarbon chains. Outside the rafts, kinks in unsaturated hydrocarbon chains and lower cholesterol concentration result in increased fluidity. At sites of inflammation, secreted chemokines bound to heparin sulfate autoglycan on endothelial cells are presented to leukocyte 7 transmembrane receptors. The binding stimulates leukocytes and triggers an intracellular cascade of signaling reactions. The inner leaflet of the bilayer is a very different composition than that of the outer leaflet. While some proteins traverse the membrane, others are either anchored into the inner leaflet by covalently attached fatty acid chains or are recruited through non-covalent interactions with membrane proteins. The membrane-bound protein complexes are critical for the transmission of signals across the plasma membrane. Beneath the lipid bilayer, spectrin tetramers arranged into a hexagonal network are anchored by membrane proteins. This network forms the membrane skeleton and contributes to membrane's development and membrane protein distribution. The cytoskeleton is comprised of networks of filamentous proteins that are responsible for the spatial organization of cytosolic components. Inside microfilament, actin filaments form tight
Best protein ever.
which are stabilized by cross-linking proteins. While deeper in the cytosol, the actin network adopts a gel-like structure, stabilized by a variety of actin-binding proteins. Filaments, gapped at their minus ends by a protein complex, grow away from the plasma membrane by the addition of actin monomers to their plus end. The actin network is a very dynamic structure with continuous directional polymerization and disassembly. Severing proteins induce kinks in the filament and lead to the formation of short fragments that rapidly depolymerize or give rise to new filaments. The cytoskeleton includes a network of microtubules created by the lateral association of protofilaments, formed by the polymerization of tubulin dimers. While the plus ends of some microtubules extend toward the plasma membrane, proteins stabilize the curved conformation of protofilaments from other microtubules, causing their rapid plus end depolymerization. Microtubules microtubules provide tracks along which membrane-bound vesicles travel to and from the plasma membrane. The directional movement of these cargo vesicles is due to a family of motor proteins linking vesicles to microtubules. Membrane-bound organelles, like mitochondria, are loosely trapped by the cytoskeleton. Mitochondria change shape continuously, and their orientation is partly dictated by their interaction with microtubules. All the microtubules originate from the centrosome, a discrete fibrous structure containing two orthogonal centrioles and located near the cell nucleus. Pores in the nuclear envelope allow the import of particles containing mRNA and proteins into the cytosol. Here, free ribosomes translate the mRNA molecules into proteins. Some of these proteins will reside in the cytosol. Others will associate with specialized cytosolic proteins and be imported into mitochondria or other organelles. The synthesis of cell-secreted and integral membrane proteins is initiated by free ribosomes, which then dock to protein translocators at the surface of the endoplasmic reticulum. Basic proteins pass through an aqueous pore in the translocator. Cell-secreted proteins accumulate in the lumen of the endoplasmic reticulum, while integral membrane proteins become embedded in the endoplasmic reticulum membrane. Proteins are transported from the endoplasmic reticulum to the Golgi apparatus by vesicles traveling along the microtubules. Protein glycosylation, initiated in the endoplasmic reticulum, is completed inside the lumen of the Golgi apparatus. Fully glycosylated proteins are transported from the Golgi apparatus to the plasma membrane.
Glycosylated.
When a vesicle fuses with the plasma membrane, proteins containing the vesicle's lumen are secreted, and proteins embedded in the vesicle's membrane diffuse in the cell membrane. At sites of inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G-protein-coupled membrane receptors. This binding causes a conformational change in the cytosolic portion of the receptor, and the consequent activation of the subunit of the G-protein. The activation of the G-protein subunit triggers a cascade of protein activation, which in turn leads to the activation and clustering of integrins inside lipid rafts. A dramatic conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the Roman leukocyte at the site of inflammation. Additional signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte. The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the 4 steps of a process called leukocyte extravasation.
Cool, right? Okay, like the one thing that I hate about this model, but it wouldn't work unless they did it this way. Do you notice that like every little thing that they were highlighting looked like it was by itself? It was all alone. It's crap. The cell is so packed. It is like full to bursting of stuff. So nothing happens like by itself like that. Have you seen the pictures? I don't remember what it's called. I keep saying that one of these days I'm going to get one for my office, but it's like the diagram inside. nerve cell, but it's that's not it. So not it, but it's the real. It's real. But it's like so packed, and it's every single molecule. It's a beautiful, beautiful diagram, but I don't see it now. But like, it's so packed, it's so full of stuff. It's absolutely insanity. But the stuff doesn't occur really like that by itself. All right. So if you have any questions about your exams, y'all can ask me, y'all can go, but just remember exams come here before you leave, and don't forget to come and grab your Francis. Hunty, I'm sorry. I'm sorry. Your case study. I got distracted there. Hunter, this is yours. But if you don't have any questions, I'm happy to answer them. I'm just gonna turn the thing off. And if you were out today, you can come see me and I'll give you yours. All right, have a great rest of your day.